Okay, this video is on mitosis and meiosis. These are two very important processes that your cells undergo, and they are used for different things at different points in your life. So mitosis is the process by which a single cell makes two exact copies of itself, and meiosis is when a single cell actually reduces the number of its chromosomes and makes sort of a half a copy of itself. So it goes from diploid to haploid. Now, both these processes are particularly important to you. At some point you looked like this picture up in the left-hand corner here, and you are on the planet because your parents underwent meiosis to produce their gametes, which led to you. And you don't look like this anymore because these cells underwent mitosis to become the multicellular organism that's sitting here watching this video right now. So before we talk about those processes, we need to go over a couple of terms, a couple of sort of basic principles. And the first is defining what a chromosome is. So the chromosome is actually pictured over here to the left. It is a single hot dog structure. So I'm outlining a single chromosome. This is actually a duplicated chromosome right now, and I'll point what that structure is called out in a moment. But basically a chromosome is condensed DNA. So if, over here to the right, you can see this familiar structure of DNA, and it is wrapped around these little purple protein balls called histones. And so these histones allow it to wrap, and then those histones coil around until you get the condensation. Now, you've heard about this before when we talked about differential gene expression, and we were talking about acetylation and methylation, and how both of those affect how tightly wound the DNA is around the histones, and how that affects differential gene expression. Now, humans have 46 chromosomes, so you can see here these pairs are numbered 1 through 22, and then we have a pair of sex chromosomes. Okay, so the sex chromosomes in humans are X and Y, and all these others, the 22 others, these are called autosomes. Difference is that the sex chromosomes differ between males and females. The autosomes do not. So if you were just looking here at chromosome pair number 1, you wouldn't be able to tell if this individual was a male or a female. Okay. Now, in humans, we have X and Y. You should be able to tell what sex this individual is. It is a male. Females have 2 X chromosomes. Now, other species have different sex chromosomes. Sometimes the female is actually the heterogametic sex, meaning that it has 2 different sex chromosomes, and sometimes they're not called X and Y. So birds, for instance, the females are ZZ, and the males are ZW. This involves the differences in the sex chromosomes and how they evolved, which is pretty fascinating. In addition, other species do not necessarily have 46 chromosomes, so you need to be careful about being so human-centric when you're thinking about these things. I often refer to our 46 chromosomes just to touch base with the concept, but fruit flies, for instance, only have 4 chromosomes. And so there's a lot of variation, and oftentimes when you're looking at pictures of mitosis and meiosis, they only depict a situation in which there are 4 chromosomes, simply because it would be really difficult to draw 46 in a diagram in your book. Now, you've got 2 copies of each type, okay? And if you guess where they come from, one set comes from your mom, and one set comes from your dad. So, for instance, these 2 chromosomes, unless, you live in the South, should not be identical, right? And that's an awful joke. But really, they shouldn't be identical because they came from 2 different individuals. Okay, yet they are the same in a sense. because they are chromosome 1, so they contain all of the same genes. So we call these not identical chromosomes, but rather homologous chromosomes. Okay, so homologous doesn't mean the same, it means sort of the same. They have the same genes, but they possibly differ in their sequence. So the version of each gene that you find on these chromosomes might be different. So when you kind of reflect back to high school biology and Mendelian genetics, these were the big P's and the little p's, right, that you dealt with those pea plants. The big P is one version of a gene and the little p is another version, but they still are a gene that code for petal color. Okay. So you can see these pink genes up here will code for the same trait, although the sequence might be different because they came from two different people. Now, like I said at the beginning, okay, mitosis is used for growth, development, and repair, and usually occurs in what's called the somatic cells. So somatic meaning body cells, so like your skin cells, your blood cells, your muscle cells, that sort of thing. And meiosis is for gamete production, so these are your reproductive cells. They're the sperm and the eggs, so they occur in your sex organs specifically. So in female humans we have ovaries in which our eggs are produced, so eggs are female gametes, and males have testes in which their sperm is produced, so sperm are male gametes. We're going to talk about mitosis first, although both processes are very similar. Okay. So if we're thinking about different cells, okay, the division rates, the rate at which those cells multiply, can vary. Okay, so for instance, your nerve cells, most of them never divide. So they never undergo mitosis once you reach adulthood. While other cells, like your skin cells that line your stomach, or your skin cells on your arm, for instance, they divide very rapidly. So, the oldest skin cell you have might be 3 days old, right? So that's really interesting. Regardless, though, once a cell undergoes the process of division, they all follow the same cell cycle. Okay, so this picture is the cell cycle. Okay. We're just following a single chromosome. This just represents a diploid cell. So diploid, if you're imagining your diploid cell, it would have 46 chromosomes in it. Majority of the time, the cell is in interphase. So this is a phase where the cell is just sort of chilling out. It's sort of preparing to divide. Okay, so as it's going around the cycle, there are a couple of checkpoints, which we'll talk about later. So as it goes around and around, it's going through different changes. At some point, it has duplicated its chromosome. So all cells, whether they're going through mitosis or meiosis, will undergo this initial stage of chromosome duplication. They'll make an exact copy of themselves. So you can see that's this little X structure that was in the very first picture I showed you. And that X structure, these represent duplicated chromosomes. So these are often called sister chromatids. Okay, and they are held together at a centromere here. Now those chromosomes, those duplicated chromosomes, finish the interphase and then will undergo active mitosis. So this is the mitotic phase. So that cell now has duplicated DNA, will eventually divide into two different cells. So you can see one cell here and one cell here. That splitting off into two cells is called cytokinesis. Okay. So there's a lot that's happening in both interphase and mitotic phase. So we're going to take a look at that. Mostly we're going to focus on mitotic phase. So I've said both of these things. Alrighty, so the importance, the why cells duplicate, it's to conserve the number of chromosomes. So if you think about your diploid number of chromosomes, wanting to make an exact copy, okay, you can't just divide your chromosome or your cell in half. If you did that, then each of those new cells would only get half the number of your chromosomes. So duplication always conserves the number of chromosomes, which is important for organism survival. Okay, so you can see, again, the terminology that I've used. We've got this single chromosome, which is the hot dog shape. Once it's duplicated, okay, this is called sister chromatids, and they are attached always at the centromere until they are getting ready to divide. Okay, so when they divide, then they become chromosomes again. Note that the sister chromatid structure is different than homologous chromosomes. Whenever we're referring to homologous chromosomes, generally they are not connected. They're just side by side because they're similar. Sister chromatids are connected in sort of this X structure, so it looks like an X to me. I don't know if it looks like an X to you or not. Alrighty, so let's look at the steps in mitosis. So this first step here, this is the interphase. This is where the cell is getting prepared to undergo mitosis. So at this point, all of the DNA is sort of loose in the cell, and it's beginning to condense into chromosome structures. So it's wrapping itself around those histones, getting ready to become those visible hot dog things. And so you can see what these cells actually look like in the pictures above, which is pretty neat. Before the cell goes into the mitotic phase, which is the first phase of the mitosis is prophase, remember the DNA has duplicated. Okay, so in this picture, when we see our chromosomes, they are in our little familiar X shapes, and so these duplicated chromosomes here are attached together, the sister chromatid. Okay. So you might want to pause and test your understanding and ask yourself, if we're looking at prophase, we're looking at this guy here, okay, assuming this isn't a human cell, what is the diploid number of cells that this individual or species has? It's going to be helpful, okay, you don't want to just go through and memorize the steps, you actually want to think about, okay, what did we begin with? Where are we in the process? Are we duplicated or are we not? And then what is the end goal? What are we ending with? And make sure that you're sort of keeping track of the chromosome number in order to understand what's happening at each step. So again, those represent duplicated chromosomes, and that duplication happens before prophase. Now in metaphase, those duplicated chromosomes are lining up along the equator of the cell, and they're lining up in a straight line. That is really important because when we look at meiosis, they don't line up in a straight line at first, and that has really important consequences for what's going on in mitosis at this point in the process. Okay, so when I say they line up end to end in a straight line, I mean the little tip, okay, the top and the bottoms are all sort of lined up end to end. When I say side by side, oops, I'm talking about this side here. Okay, so this is the side of the chromosome, and these are the ends. That's what the terminology I use. So once those are all lined up end to end, they actually are separated. The sister chromatids are separated, and the chromosomes are pulled along here, the cytoskeleton, to the poles over here. I'm sorry, I'm not drawing, I'm not making very good doodling. But they're each half is pulled over to one side. Okay. Once that happens, then the cells undergo cytokinesis and they separate. Okay, so at this point, because we're dealing with mitosis, which again, remember, is the process by which we're making two exact copies, we've got two cells that have all of the DNA. Okay. So we're going from one diploid cell to two diploid cells. Now, as I sort of alluded to before, mitosis has checkpoints. This is an actually highly controlled process. It has to be, right? Your cells, first of all, have to sort of, you don't really realize, but I'm going to use that language, your cells have to realize what kind of cell they are and undergo mitosis at an appropriate rate. Okay, so again, your skin cells divide faster than your nerve cells do, right? And so that is controlled by checkpoints that are in each of your different cells. Now, in general, mitosis has at least 3 checkpoints. Have one, so what we're looking at here, this M phase, that's the mitotic phase, and then this around here is all the interphase. So just before the cell is going to duplicate, it has a checkpoint, so it needs to make sure that everything is okay before it starts to duplicate its chromosomes. Okay. You might think for a moment about why that's very important. Everything should check out before we do that very important process of making copies of our DNA. So once that happens, then the cell undergoes another checkpoint here, so there's a G2 checkpoint before it begins mitosis, and then it goes through another checkpoint after mitosis. And so again, both of these checkpoints are checking for something slightly different than the G1 checkpoint. There are also external checkpoints. Okay, so certain cells know when to stop growing when they are exposed to different environmental factors. So one really common one is crowding. You put a cell in a petri dish, it will divide until it forms a layer of cells along the bottom of the petri dish. Once it starts feeling the pressure from nearby cells, mitosis will be shut off. So this is an external checkpoint, whereas the internal checkpoint happens inside the cell. So I just described the space. Also, cells, when they grow, they need nutrients. So if there's a limited amount of nutrients, this could be a good sign to stop growing as well. Okay, so we're going to switch and talk about meiosis. Meiosis is a process that occurs in sexually reproducing species. So it usually involves 2 gametes. We've got egg and sperm here. These guys will unite in a process called fertilization to produce a zygote, which will eventually become an embryo and eventually become a multicellular organism. Now, in terms of what's going on inside at the chromosome level, our gametes have half the number of chromosomes that our zygote should have. So in humans, each egg and sperm cell have 23 chromosomes, and that will come together to make a 46 chromosome zygote. So diploid number, full number of chromosomes, is 46 in humans. Remember, it's different numbers and different things, and in order to get that, we are undergoing sexual reproduction. You have to have 2 gametes that have half that number. If you imagine each of these gametes having 46, when they come together, that would be 92. and that would not be human then, right? Development wouldn't proceed normally. So as you might guess, it's very important that meiosis happen in an appropriate manner, that the chromosomes will duplicate and the cells will separate in a very orchestrated manner. Alrighty, so meiosis actually consists of two sort of steps, I suppose, meiosis 1 and meiosis 2. And each step has different phases that are the same as the phases we went over for mitosis. Okay, so prophase is that first meiosis phase. Remember, we've already gone through interphase, and so our chromosomes have already been duplicated and condensed. Okay. So in prophase, something else happens. So in addition to having our chromosomes duplicated, once we're in prophase, the duplicated homologous chromosomes will line up side by side, and they exchange genes. And this process is called crossing over. So you can see here these two long red chromosomes. These represent duplicated chromosomes, say from your dad, and these blue chromosomes here represent duplicated chromosomes from your mom. Remember, these guys carry the same genes, they're very similar genes, so they're homologous. They find each other, they line up side by side, and they make an equal swap. So you can see that now this blue chromosome here has a little red tip, and this red chromosome here has this blue tip. It's very important to make an equal swap. If you don't make an equal swap, then one chromosome will have more genes than it should, or fewer genes than it should, and that will lead to problems down the road once we have separation of these chromosomes. Now, once that happens, the chromosomes line up along the poles in metaphase. This time, however, instead of lining up end to end, these homologous chromosomes stay together and they sort of line up, well, they're lining up side by side, but you can see that they're not all in a straight single line, but they're sort of in this double line. Okay? And the way in which they line up is random. Okay. So you can see, for instance, that not all of the red chromosomes lined up on the bottom and all the blue chromosomes lined up at the top. They just sort of sort themselves randomly. And you can imagine that if you have all of your chromosomes doing this, right, you have all 46 lining up this way, there are lots of possible combinations for which they can line up along the equator. I'm making such a big deal about that because the random alignment of chromosomes here at metaphase and the crossing over, okay, both of these will create diversity in our gametes. So you'll see that at the end, gamete isn't all blue chromosomes or all red chromosomes. It's a mixture. It's a shuffling of your mom and your dad's genes. And that's because of these molecular processes that are going on here, these cellular processes, right? Okay. So what's really cool, two individuals can actually create 6 billion different looking kids if you kind of figure out all the possible combinations of their gametes. There's a large diversity being produced, and that's because each time you undergo meiosis, there's so many different ways to cross over, and there are so many different ways to randomly align up. Alrighty, so the cell then undergoes anaphase, which is the splitting or the pulling apart of these duplicated sister chromatids. Now you'll notice in this anaphase, the sister chromatids aren't separated yet. The homologous chromosomes are. So the sister chromatids are still attached, and the anaphase is bringing apart all 4 of them to each side. Okay. So we're undergoing cytokinesis again. At this stage, telophase 1, we still have our sister chromatids. Now they're just shuffled up. Okay. So this first division yields a diploid number of chromosomes. If you kind of go back and figure out the number that we began with, you can see that at this phase it's the same number. So 4, right? Alrighty, so now on to meiosis 2. So not much different happens in prophase 2. There is no crossing over or anything, and there is no duplication of DNA. We've already got that over and done with. The real meat here is metaphase 2. So in metaphase 2, these sister chromatids are lining up end to end, okay, just like in metaphase in mitosis. And that should make sense because if you think about what we want to make in meiosis, we want to make haploid gametes. We're going to get ready to separate these here. And what you can see is that at each cell, if you imagine this divided in half and splitting over here in telophase, each of these new cells is now going to get half the number of chromosomes. So we started with 4. That was our diploid number, and now we have 2. So we started with a single diploid parent cell, and now we have 4 haploid daughter cells, which are quite different genetically from our parent cell. So it's a combination of both of the parents. So again, you just want to keep track that in meiosis 2, we've got our chromosomes lining up in a straight line because we want that final separation. Now this picture here is just comparing and contrasting the 2 processes. Okay, so the first prophase, metaphase, anaphase, and telophase are to some extent very similar. We're going to start up here at the top. We've got a parent cell, okay, that has 4 chromosomes. That's our diploid number, so 2N means diploid. And we know that this is before replication, before duplication, because we've got our hot dog structures rather than our little X structures. Okay, so chromosomes duplicate, and now we have 4 little X structures, right? So this structure here is a sister chromatid, right? Sister chromatids. Now in meiosis, remember we have crossing over. So we have our duplicated homologous chromosomes finding each other, and together these guys are called a tetrad because there are 4 chromosomes over there. So that tetrad is essential in crossing over and exchanging those genes. Now in mitosis, metaphase, we've got them, all the chromosomes lining up end to end. Here in metaphase 1, over here in meiosis, we still have those tetrads lining up, and we've got side-by-side alignment. In anaphase, we've got them separating, and in telophase, we have them, the cells actually pinching off to make daughter cells. So in mitosis, at this point, we're done. In anaphase, each of the each complement of chromosomes is pulled to either side, and when we split off, now we have daughter cells that look exactly like our parent cell. Okay. So that's necessary if we want something to grow, right? Or we want to heal a wound, for instance, and we want to make new skin cells to heal up that wound that we have. We need to make sure that those cells are actually exactly the same. In meiosis, when we get to anaphase and telophase down here, we're actually not done, right? Remember, the whole goal of meiosis is to have a haploid cell, okay? In order to do that, we need to get down to 2 chromosomes, and so right now we still have 4 chromosomes in these cells. We still have these sister chromatids attached. So they also need to reline up again, and then they split. So they're like lining up like this metaphase over here. They're splitting, and they're making 4 daughter cells, okay, which are all haploid. So the N by itself means half the number of the diploid. And if you compare these cells, indeed they have 2, whereas that original cell had 4. And you can see also that these chromosomes are quite different than any that are found in the parent cell, and that's the diversity that I was talking about earlier. When you read about sexual reproduction, you often read about how it produces diversity, and you've already learned about 2 ways that occurs within meiosis. A third way in which diversity is created is when these, if these are egg cells, these egg cells will randomly pair up with a sperm cell, right? So that random pairing shuffles the genes even more. But be careful, that is a feature of sexual reproduction, sort of at a large level. It isn't a feature of meiosis, right? We don't have any sexual reproduction happening within meiosis. This is the process that has to occur before sexual reproduction happens. Okay, so key ideas. All cells have to arise from existing cells, and that happens through the process of mitosis. Mitosis is the process by which cells are replaced or repaired. So damaged cells have to repair themselves. When you get a wound, you have to heal that wound. But also mitosis is used during development. So when you are that little ball of cells and you need to get bigger and bigger, mitosis is happening within each of those cells. Meiosis makes gametes, so this allows organisms to undergo sexual reproduction, and the series of duplications and divisions that happen within this process allows the species to conserve their chromosome number, which is important if you want to make a baby that actually functions. They have to have the correct number of chromosomes to do that. In meiosis, we get this diversity through crossing over and random alignment of chromosomes in metaphase 1. Now, it's important to remember that it's important that both of these processes happen very precisely. Okay? That doesn't mean, however, that there aren't errors. Okay? These processes are prone to error. So the whole act of duplicating your DNA, there might be an error during that process. There might be an error as cells are dividing. There might be an error in terms of crossing over or alignment of cells. And so when there are errors, that leads to issues. For instance, in mitosis, when you have an error in DNA duplication, for instance, that's what we call a mutation. Right? So if the DNA doesn't duplicate properly, you get a nucleotide that's not supposed to be there, or you get, a whole gene that's copied, that's already been copied. That is a mutation. And most of those mutations will kill the organism or the cell or whatever it happened in, but some of them do get through and might have a beneficial effect or not for the organism. In meiosis, the process should happen correctly, or else you get birth defects, right? So Down syndrome, for instance, is caused by an additional copy of chromosome, I believe 21, and that is because there was an error in meiosis. Those chromosomes didn't divide properly, and you have one set of gametes that has more than it should have. Okay. So in general, errors in mitosis and in meiosis happen as you age. So as you age, your cells start to break down. The mechanisms, the checkpoints aren't as good as they used to be, and these things are prone to more error. But we're going to talk about that in more depth when we talk about cancer and aging later on.